This video will be the first of the genetics series, and in genetics we are going to go over all of how DNA is actually structured, how it's replicated, and how is it actually used, how are the genes that are encoded in that DNA used to build proteins. So far we've talked about, first semester, the DNA is duplicated before the cell divides, so we've talked a little bit about obviously how important the DNA is. You've heard that that the nucleus that holds the DNA is the control center of the cell. Well, in this unit we'll actually find out once and for all what is the critical function of the DNA, the instructions for all the proteins that build us. I like to start this unit by giving students a real quick background on a couple of experiments to give students an idea of the really rapid change in our understanding of DNA. Even back in 1928, we didn't know the function of DNA. If you look at this experiment, this very famous Griffith's experiment, what he did was he took bacteria that appear smooth under a microscope, and when he injected them into a mouse, they were always lethal to the mouse. And he took other bacteria that were rough under the microscope, and when he injected them in the mouse, they didn't harm the mouse. So he had some bacteria that were lethal, other bacteria that weren't. And what he did, so his first step, was he took that lethal strain of bacteria, he heated them to kill the bacteria, and when he injected it into the mouse, it didn't hurt the mouse, which should make sense to you, obviously. Even if they're lethal bacteria, if you've killed them, they're not going to be able to harm the mouse. But then he did something interesting. He took the lethal bacteria, heated it and killed it, and then he mixed in some of these non-lethal bacteria. So after he'd heated and killed these lethal ones, he then mixed that solution with these non-lethal bacteria, and then he injected that into the mouse, and the mouse died. And that was a very interesting result, because what it told him was that somehow these non-lethal bacteria had been transformed. That was the word that he used, transformed into the lethal bacteria. Now, he had no idea what the cause of that transformation was, but something had come from this lethal bacteria to the non-lethal bacteria, changed them into lethal bacteria. And indeed, scientists disagreed about what it was in the bacteria that had actually caused that transformation. And most scientists at the time actually believed that it was probably the proteins, because the diversity of proteins was beginning to be understood, and it was thought that genetic information was likely carried in proteins. But it wasn't until 1944 when the deciding experiment was done. And what this experiment was essentially a replication of Griffith's experiment, except for instead of taking the entire smooth bacteria, they took pieces from the smooth bacteria. So they took proteins from the lethal bacteria, mixed it with the non-lethal bacteria, and the mouse didn't die. They took lipids from the lethal bacteria, mixed with the non-lethal bacteria, and the mouse didn't die. But when they took DNA from those lethal bacteria and mixed it with the non-lethal bacteria, then that's when the magical transformation occurred. That's when the rough cells changed into the smooth lethal cells and killed the bacteria. And this experiment demonstrated once and for all that, in fact, DNA was really that magical molecule that was actually coding for the different traits. In this case, the smoothness versus roughness of the bacteria, and whether it was lethal in the mouse or not. So that experiment showed that DNA was the molecule that actually carries heredity. It was the molecule that transformed this bacteria. And really, that's back in 1944, which might sound like a long time ago, but really it's not. So it's kind of amazing to think about that back in 1944, scientists were just beginning to understand the importance of this DNA molecule, and to see the types of things that scientists are able to do with DNA today. We now have a much better understanding of how the information that's encoded in DNA, we'll call that information genes, how those genes are actually used to code for protein structure, and we'll learn this two-step process of transcription and translation in this unit. So this particular video is going to be the basics of the structure of DNA and RNA, structure and function. So DNA carries the genetic, which is the heritable information, which is responsible for passing traits from one generation to the next, from the parents to the offspring, whereas RNA, we'll learn there's actually 3 different types, and you should probably write this somewhere in your study guide, just so right off the bat we know that there's 3 different types that you'll need to know about from my class. There's mRNA, there's tRNA, and there is rRNA. And these 3 different types of RNA are all used in the process of making proteins. They're all going to have specific roles in that process of transcription and translation. So you have this picture on your study guide and also on the screen here. I'd like you to pause the video and I want you to try to identify the differences between the DNA that you're seeing over here and the RNA that you're seeing over there. And I want you to identify, there should be 3 that you can find. So look at this picture over here, compare it over there, what's the differences? So pause the video and do that. The first difference is highlighted right here. The first difference is the type of sugar that's used. In DNA, the sugar is a deoxyribose sugar, whereas in RNA, it's a ribose sugar. And we'll talk about what that means in just a moment. That's the first difference. The second difference is highlighted there and there, that we've got in DNA, we use 4 bases, adenine, cytosine, guanine, and thymine, abbreviated as A, C, G, and T. But in RNA, we use adenine, cytosine, guanine, and uracil. So it's A, C, G, and U. So that notice the A, C's, and G's are the same, but in RNA we replace the thymine with the uracil. That's the second difference. And the third difference, which should be hopefully fairly obvious, is that if you notice DNA is double-stranded, right? There's 2 strands, and RNA is single-stranded. So you need to know those 3 differences. You know the sugars are different, the bases are different, and DNA is double-stranded, RNA is single-stranded. You will need to know for my class what the letters DNA actually stands for. DNA stands for deoxyribonucleic acid. And the nice thing about that name is that it tells you exactly what it is. It tells you it's a nucleic acid. That's one of our 4 polymers made from nucleotide monomers. So if you remember that from way back in Chapter 2. But it also tells you that it's using a deoxyribose sugar. So that's why it's called deoxyribonucleic acid. Whereas RNA stands for ribonucleic acid. So it is not using a deoxyribose sugar, it's simply using a ribose sugar, and hence, as a result, it's called RNA or ribonucleic acid. Nucleic acids are polymers made from nucleotide monomers. This slide is literally copied and pasted from way back in Chapter 2 when we learned this. Remember polymers, we had our 4 polymers, lipids, carbohydrates, proteins, and nucleic acids. You had to know the monomers that build them. For nucleic acids, they're made from nucleotide monomers, and we also mentioned the fact that nucleotides themselves are actually fairly complex molecules. You have 3 different sections of sugar, which we just covered is going to be a deoxyribose sugar or ribose sugar. It's going to have a phosphate group, a negatively charged phosphate group, and then one of 5 bases, A, C, G, T, or U. Out of the 5 bases, each of our different types of nucleic acids, whether it's RNA or DNA, is going to use only 4. So they all, both DNA and RNA, will use adenine, guanine, and cytosine. The difference is that in DNA, we use a thymine, and in RNA, we use a uracil. Now, let's talk about what this difference is between ribose sugar and deoxyribose sugar. If you look at these 2 sugars, you'll notice that they're both ribose sugars, but there's something different about the 2, and it is highlighted, so I'm hoping that you're noticing it. If you notice, this ribose sugar has an oxygen and a hydrogen here, whereas this ribose sugar only has the hydrogen. It has been deoxygenated, hence deoxyribose. So a deoxyribose sugar is just a ribose sugar that has been deoxygenated right there. Okay, so that's when you say deoxyribonucleic acid or ribonucleic acid, what you're saying is that it's going to be a nucleic acid built from one of these 2 different types of sugars. This sentence is perhaps the most important discovery that human beings have ever made. What this slide says is that when it comes to nucleotides, the part that really matters, the part that's actually going to give meaning to the nucleic acid is the sequence of the bases. So it's not the sugar it's used, it's not the negative phosphate, and in fact, to give you an idea of how unimportant those parts are, in most diagrams of DNA they're just shown as this kind of ribbon. So we're not even illustrating the sugars and the phosphate backbone, we just know it's there. What's important is the sequence of the bases. It's these letters, the sequence of those letters is what's actually going to give this molecule meaning. So G, T, T, A, T, G, C codes for something. And it codes for something different than G, C, T, A, C, G, T, T, A. And what's really profound is that the code that's been deciphered by scientists is universal, meaning that this sequence means something, and it doesn't matter if I pull that sequence out of a human cell or a plant cell or a bacterial cell. I can tell by reading the code what that code is actually coding for. So this is a truly amazing discovery that scientists have been able to make and understand, and the stuff that we've been able to do with that information has been really mind-blowing. So this code is true for all species, and the amazing part is it's used the same in all species. So what can scientists do with that? Well, the fact that the code is read and used the same in all species allows for scientists to take genetic information from one organism and put it into another organism, and it will work. And we call those kinds of experiments genetic engineering, and we call the kinds of organisms that we create doing that genetically modified. And it's those types of things that are really creating some pretty amazing advances. So the rest of this video will be talking about the actual structure of DNA itself, this molecule of DNA. DNA, as we just looked at a second ago, is double-stranded, and the two strands are held together by these fairly weak bonds called hydrogen bonds. What's a hydrogen bond? Well, what a hydrogen bond is sort of like a magnetic attraction that happens when a slightly negative region of one atom comes in contact with a slightly positive region of another atom, and they attract together like magnets. If that's sounding familiar to you, I'm hoping it does, because it's actually those hydrogen bonds that form between water molecules that makes water such an amazing. Remember, water has a slightly positive hydrogen end, has a slightly negative oxygen end, and those negatives and positives interact, right? It makes the water molecules behave like tiny magnets. So it's pretty amazing that the same bonds that are so critically important for why water is so important are actually the exact same kind of bonds that are holding together my double helix. Now, one thing you should make a little note to yourself on your study guide, I want you to explain this idea. The reason why this is such an elegant system is that each of these individual bonds is fairly weak, meaning that to unzip the DNA double helix doesn't take an immense amount of energy. These bonds are not really hard to break in that sense. However, at the same time, because there are so many bonds, overall the DNA molecule is in fact very stable and strong. One kind of good analogy for it is Velcro. If you don't know how Velcro works, it works something like this. You have two pieces of Velcro, and on one side you've got a bunch of loops. And on the other side, the other piece of Velcro are actually a bunch of little tiny hooks. And when you push those two together, what happens is a whole bunch of the little hooks end up in little loops. Now, each one of those interactions is very weak. But when you have an entire sheet of it pushed together, all those little interactions make it very strong. So, why this is such a beautiful system is it allows for the DNA molecule to be very usable. You can unzip it fairly easy, but at the same time, overall, the entire molecule is very stable. Now, the DNA double helix, we know, is what's called antiparallel. Okay, what does antiparallel mean? Well, we should know from our math class that parallel means that the two lines have the same slope. The whole idea of anti means that one strand is going in one direction and the other strand is going in the exact opposite direction, hence antiparallel. So they're parallel but going in opposite directions. If you look here in this picture, you see that the sugar phosphate backbone, which again they're just kind of showing as a ribbon, on one strand it's going from 5' over here to 3' over there, while the other strand is going in the exact opposite direction. The 5' end is here, the 3' end is there. Which is what your notes say here, that one strand is oriented from 5' to 3', while the other one is oriented from 3' to 5'. So they're parallel but running in opposite directions. Now, what in the world does that mean, 3' and 5'? Now, to really understand this, this is kind of a chemistry, organic chemistry thing, so it might be a little challenging for you, but I'll explain it. I'm hoping that you get it. Remember, each one of these is a nucleotide. Let's look at this one over here. A nucleotide is a sugar, a phosphate group, and a base. So this right here is a nucleotide. There's another nucleotide with that sugar, phosphate, and base. There's another nucleotide, and there's another nucleotide. So these are 4 nucleotides lined up, and the same is over here. Okay? Now, a ribose sugar, right, it's a sugar, phosphate, and the base. A ribose sugar happens to have 5 carbons. It's a 5-carbon sugar, just like how glucose is C6H12O6, has 6 carbons. A ribose sugar has 5 carbons. And notice that they're numbered here, 1, 2, 3, 4, 5. So you can think of this nucleotide as having a 5 side and a 3 side. the phosphate is attached to the 5th carbon, and the hydroxyl group, the OH, is actually attached to the 3 carbon. So if you notice the overall strand, there's a phosphate on one end attached to the 5' end of one ribose sugar, and that hydroxyl group is all the way down here on what would be the 3' end of that ribose sugar. And if you look over here on this strand, you'll notice it is exactly like this strand, only as if you will flipped completely upside down, right? So it's as if it's just, flipped and going the opposite direction. Now the 5' sugar is over, I'm sorry, the 5' carbon is right there on the sugar, so that's where the phosphate group is. They're showing it up here, but realize it'd be the same here. And the 3' end that has the hydroxyl group is up here. So that's what we mean by antiparallel. Now that's going to have an important consequence when it comes to how we actually add nucleotides. Because what we're going to see is that you can only add nucleotides on one of these two ends. So that's going to be important when it looks at DNA replication. Another illustration of DNA. Got your DNA double helix here, and then you're just going to show you more details there. There's your sugar phosphate backbone. So you'll realize this is the 3' end with the hydroxyl group. That'd be your 5' end up here with the phosphate. Whereas down here it's the phosphate group over here, so there's the 5' end down there, and the 3' end up there, and then you see your hydrogen bonds holding together this double helix. Last thing you need to know from this video, and it is something absolutely fundamental, is something called Chargaff's pairing rule. What Chargaff's pairing rule says is that the adenine in DNA bonds to a thymine, and the guanine in DNA binds with the cytosine. The A binds with the T, and the C binds with the G. And believe it or not, that was actually first demonstrated by this scientist, Chargaff, doing experiments where he just looked at the amount of bases that he saw in these different organisms. And what you'll notice when you look at the data is that the number of adenines matches up very nicely with the number of thymines, and the number of guanines and cytosines match up, right? And that is, we now know, because for every adenine on the DNA, it's bonded to a thymine, so the numbers should be basically the same, and the same thing with the guanine and the cytosines. And that's what's shown in this picture again. If you look at it, you'll notice that the guanines and cytosines are attached together, the adenines and the thymines, right? The A's and the T's, and the C's and the G's. So that's what Chargaff's pairing rule says, is that for every A you're going to get a T, for every C you're going to get a G, because that's how the molecule actually is put together. So knowing that, you should be able to answer this question, I'm hoping. It's just a math question. What it's saying is that if adenine makes up 20% of a segment of DNA, what percentage of DNA will be made of thymine, cytosine, and guanine? So just pause the video for a quick second and try to do that. So I'm assuming that you've given it your best shot. Here's how I solve a problem like that. I say, okay, adenine makes up 20%. That tells me automatically that my thymine must also be 20%, because for every A I'm going to have a T. Now then, what's 20 plus 20 is 40%. So 40% of my DNA is already spoken for between my A's and my T's. So that leaves me with 60% left over. And how is that 60% going to be divided up between cytosine and guanine? Well, it's going to be perfectly even. And so half of 60 is 30. So that tells me that 30% of my DNA is going to be cytosine, and 30% is going to be guanine. And then you have this final practice problem, which you should be able to pause the video and do. This time we're starting off with guanine, making up 35%. So pause the video and try to give that a shot. Okay, I'm assuming you've given it a chance. So guanine is 35%. That tells me that cytosine must also be 35%, since those are matched up, the C's and the G's. And 35 plus 35 gives me 70, so that leaves me with a percentage of 30 left over, which is going to be evenly distributed between adenine and thymine, which means 15% and 15% for those. So hopefully that video was helpful. Again, we just covered the basics of the difference between DNA and RNA and the basic structure of the DNA molecule.